Our next talk is ion mobility mass spectrometry and collision-induced unfolding for characterizing the impact of small chemical modifications on monoclonal antibodies. It'll be presented by Nicole Rivera Fuentes.
So I'm going to be talking about how we in the Rutler lab utilize ion mobility mass spectrometry to characterize how small chemical modifications can impact monoclonal antibodies. Monoclonal antibodies represent one of the fastest growing classes of biotherapeutics in the pharmaceutical industry. I'm showing here, if we look at the drugs approved only last year in 2024 by the FDA, out of the 50 therapeutic agents that were approved, 16 of these were protein-based therapeutics. And within these 16, 15 of those were either a monoclonal antibody or a monoclonal antibody-based therapeutics. And in terms of the therapeutic areas that these can target, more than half of these were for oncology applications, but other therapeutic areas such as autoimmune diseases as well as dermatology were also included. And some attributes that make MABs so successful as therapeutics include the high affinity and specificity for its antigen, as well as the low limited toxicity that they provide. However, when we compare our monoclonal antibodies to small molecules, the antibodies are significantly larger than small molecules, and this increase in size comes with an increase in the structural complexity of our molecule. Additionally, antibodies are highly dynamic molecules, which adds complexity to the structural characterization of these. And higher the structures, as well as biomolecular stability, are important quality attributes that must be monitored through their development. And not only are these intrinsically more complex than small molecules, being proteins, they're susceptible to differences in their post-translational modifications. And an example of this is their glycan composition. These are glycoproteins, and we can have a variety of differences in the chemical composition of their glycans. These are also susceptible to degradation pathways, which can affect the efficacy and safety of our product. And the two most common degradation pathways are oxidation of the methionine and tryptophan residues, as well as the deamidation of the asparagine residues. And additionally, recent development of these MAP-based therapeutics include the cross-linking of an antibody and cross-linking it to another therapeutic agent. And this further increases the heterogeneity that we can find when we're looking at these samples. So characterizing rigorous characterization of how these chemical modifications, whether it's an unwanted degradation pathway or a desired cross-linking reaction that we're targeting, seeing how these affect the stability and structure of our molecule must be monitored through the development process. And so in the Rutler lab, we utilize ion mobility to try to tackle these questions. And so here I am showing the schematic of SYNAP G2, which is an ion mobility mass spectrometer that we have in our lab. And for our experiments, the first thing that we do is take our sample from our solution, and we perform ion generation via nanoelectrospray ionization. And then these ions are separated in the ion mobility separator. And this region is filled with a neutral gas. And we have our charged particle in the presence of an electric field. So that charged particle is going to travel through the ion mobility separator. And we're going to separate our molecules based on charge, shapes, and size, where more elongated structures are going to collide more frequently with this buffer gas, and that's going to be reflected on then having a higher drift time, while more compact structures are going to collide less frequently, allowing for a faster drift time. Then we can get our mass analysis using a time-of-flight mass analyzer. And combining this data, we can generate these three-dimensional plots. And what this allows us to do is to resolve species that have the same mass to charge ratio, but differing structure. And we can also convert this drift time information into collision cross-section, which is the rotationally projected average surface area of our molecules. However, for IgGs or antibodies, drift time alone is often not sufficient to get a lot of structural information. And to overcome this, we utilize collision-induced unfolding. And so how this works, here I am showing an example of the mass spectra of an antibody. And what we can do is we can submit each of these charge states that we see in this trap region that we have that happens before the ion mobility separator. We can increase the collision voltage of our molecule, which causes the internal energy to increase and causes our protein to unfold. And we can monitor this unfolding process by monitoring the changes in the drift time. We can generate plots like the one that I'm showing. And if we look at this from the top, we can generate these fingerprints we use to visualize this unfolding process in the gas phase. And there's a couple of things that we can do to analyze this data. We can perform a feature detection where we identify those ions population that share the same collision cross-section across multiple voltages. And we can do a measurement of the CIU 50 values, which measures the point at which 50% of one of our compact states has already transitioned to a more unfolded state. And we can use this as a measure of stability, where more ions that require more voltage to reach a certain transition are said to be more stable than those that require less voltage. And finally, we can also do an RMSD analysis to see where the differences in our fingerprints are. I just wanted to mention that there were a few mass spec platforms that were utilized for this project that I'm going to show the data for, including the water-selected cyclic ion mobility, which works in principle the same way that I just explained for the SYNAPG2, as well as the Q-Exactive UHMR. And this one works with an Orbitrap mass analyzer, and it doesn't provide any ion mobility data, but it has the ability to give us extremely high resolution mass analysis. So the first project that I'm going to talk about is the analysis of an antibody-drug conjugate mimics by ion mobility in CIU. Specifically, can we analyze the effect of conjugation location on mass stability and structure? And to do this, the first thing that I did was develop some ADC mimetics that I could use as analytes. And to do this, I leveraged some chemistry available in the literature that would allow me to perform some site selectivity without the need of protein engineering. And I did this in two different ways. The first one that I'm showing in purple is leveraging the reactivity and the selectivity of a microbial transglutaminase, which catalyzes the reaction of glutamine and free amines. And for antibodies specifically, it only recognizes one glutamine in the heavy chain of the antibody. And since we have 2 heavy chains per antibody, we can get up to 2 conjugation events specifically on that one glutamine site. And another way that I used was leverage the pKa, the differences in the pKa between the amines on the N-terminus and other free amines in the antibodies, such as in the lysines. And at pH below 6, the only amines available for conjugation reactions are those on the N-terminus. And I was able to do that, to do a coupling reaction between the N-terminus and an aldehyde containing as a functional group in one of my small molecules. And I'm here showing data from the UHMR that shows that we were able to see those different conjugation events individually. After that, we moved to our ion mobility data, and I'm showing here the mass spectrum for our unconjugated MAB control and one of our reactions as an example. And we can see here that we have the same charge state distribution among the two of them, but on the water system, we needed actually a little bit of more activation than what we typically use in our experiments to be able to resolve those individual conjugation events on the MS analysis. And so, because we're interested in studying the structural impact of this, we wanted to make sure that we were not impacting our ability to see these ions in a native-like state. And so here I'm showing the control at the same conditions, and by looking at the collision cross-section values of our control under these two conditions, we see no significant differences in their collision cross-section, which means that with this method that we developed, we are able to resolve the different DARs at each specific charge states while maintaining native-like ions, which allows us to probe each conjugation event individually. And so now that we were able to do that, we moved to the stability studies, and I'm here showing the fingerprint of our control 24 plus, and we see that we get generally 3 features. So we're going to be tracking 2 CIU50s. We're going to be tracking the CIU50 for going from feature 1 to feature 2, and the one from going to feature 2 to feature 3. And so for our FC conjugated sample, we can see that we can probe from DAR 0 to DAR 2. And although this might look quite similar to the naked eye, when we get the exact values for these transitions, we see that for our first transition, there's actually absolutely no difference upon conjugation events, but we see an increase in the CIU52 value upon each conjugation event. And when we look at the map conjugated on the fab, and for comparison purposes, I'm showing as well 0, 1, and 2. We now see that the shift in stability occurs in the CIU51, while the CIU52 stays the same. And this is really exciting to us because we're looking at molecules that have basically the same molecular weight in the same amount of conjugation. We're just varying the locations of these conjugations, and we were able to see that by using CIU and looking at where this changes in stability, we can probe the location of the conjugation on the map. And changing gears now to another project. So the data that I've shown was for small molecules that were about 600 to 700 Daltons. But we were interested in pushing this a little bit forward, and so we look at degradation products, specifically oxidation, and these oxidation modifications are really small and range from 4 Dalton to 32 Dalton differences, but although they're quite small, they can have adverse effect on the map, such as alter their physical properties, disrupt antigen binding, and can result in decreased thermal stability, as well as increase the aggregation propensity of our molecule. And how we usually go about looking at this oxidation and identifying where this oxidation is happening is through peptide mapping workflows, where we take our protein, so our whole map, and we digest it down to a lot of small peptides, and this can take between 16 to 18 hours. And then we use LCMS to separate these peptides, as well as to identify their mass, and that can take between 60 to 90 minutes per sample. And then the data analysis, for IgGs, we can see up to 3,000 unique ions for each of our samples. And we have to identify these shifts in mass, so this process is quite time-consuming. And so we wanted to see if we could detect these oxidations events by using our ion mobility in CIU. So the first thing that I did forced oxidation of a model IgG4, and I performed both tryptophan and methionine oxidation by using different oxidizing agents, and by performing bottom-up proteomics, I was able to identify that these different oxidizing agents were actually hitting different amino acids. So we can see that we utilize AAPH, which I'm showing there in red. We get primarily tryptophan oxidation, specifically there are two tryptophans that are oxidized over 50%, and when we utilize peroxide TBHP, we get primarily methionine oxidation. And so this time we don't have, although we can see an overall shift in the mass, we're not able to resolve this individual as we were for the ADCs. However, by performing an analysis in the similar manner, we were able to see that for our tryptophan oxidized molecules, we saw an increase in both the CIU50 and 2, so it seemed to have an increase in the gas phase stability, while methionine oxidation resulted in a decreased gas phase stability in both transitions as well. So not only are we able to detect changes in stability produced by oxidative modifications, but we can actually differentiate if these are tryptophan or methionine oxidation. So hopefully I've convinced you that we can utilize ion mobility in CIU to detect changes in the population of antibodies as a function of small chemical modifications. Specifically for ADCs, we can identify where these conjugations are taking place. And for oxidation, we're able to differentiate between tryptophan and methionine oxidation given their distinct gas phase unfolding.